Good morning, everyone. It's a great honor to present my research at IE NanoMED. Unfortunately, due to my visa issue, I could not attend in person and can only share my work via the video presentation. The topic of my presentation is enhancing the cardiomyocyte maturation via the mechanical stimulation of the 3D printed cardiac tissue using the origami-based 3D sensor and the magnetic field. In 2019, cardiovascular diseases accounted for almost 18 million deaths worldwide. Alarmingly, mortality rates among younger population are rising, driven by the sedentary lifestyles, growing obesity rates, and the environmental influences. While pharmaceutical companies are actively developing some treatment for the heart diseases, progressing in the cardiovascular drug development continuously to lag behind other medical fields, emphasizing the urgent need for the innovative therapies. Currently, gold standard of the drug screening methods are patch clamp and microelectroarray. Those methods focus on the electrophysiological property of the cardiomyocyte, but overlook their broader physiological characteristics. Mechanical contraction force is another key parameter that reflects the cell ability to generate and sustain force, directly correlating with the functional state of the heart. Contraction force monitoring not only evaluates the cell health and functional integrity, but also aids in the studying diseases like heart failure and lung QT syndrome. Research on cardiomyocyte is evolving from single-cell studies to 2D tissues and finally to 3D tissues. Most of monitoring platforms today still focus on 2D tissues. While those methods can provide some useful data, but they cannot fully replicate the natural environment of the heart. On the other hand, 3D heart tissue are much more closer to how the heart actually works. Those tissues allow the cell to grow and behave in the way that closer in real-life conditions. Because of this, 3D tissue mode has become more and more important tool for studying heart diseases and developing new treatment. In our previous work, we developed a cantilever-based system with string sensors to measure the contraction force of a 2D cardiomyocyte. Our efforts focus on two key areas. Firstly, sensor integration, enhancing the sensor sensitivity to achieve the better detection performance. ME and string sensors are integrated on the cantilever to simultaneously monitor the electrical physiology and the mechanical contraction force of cardiomyocyte tissues. This will be explained in detail on the next slide. Secondly, the cell maturation, promoting the cardiomyocyte maturity using the conductive surface and introducing the electrical and mechanical stimulation. Recently, we built on this work to develop a high-throughput drug screening platform that synchronously measures the cardiomyocyte contraction force and the electrophysiological properties. This platform provides deeper insights into the effects of different drugs on the mechanical and electrical property of the cardiomyocyte, and the results have been published in the journal Small. We realized that while cantilever-based sensors work well for the monitoring 2D tissues, the lack of physiological relevance in the 2D mode limited the reliability of the drug screening result. This motivated us to focus on the 3D heart tissues to develop a more reliable platform for drug screening application. Many researchers have explored methods for measuring the contraction force of 3D engineered heart tissues, such as using the biofibers and elastic posts to quantify tissue contractility. However, these methods often rely on video analysis and image-based tracking, which are limited by video frame rate and motion artifacts, making real-time monitoring with high resolution and throughput challenging. To overcome this limitation, we developed a 3D sensor platform integrating ion microrod and string sensors onto the PI subject. This platform is assembled into a 3D structure by origami techniques. This platform allows us to apply the mechanical stimulation and monitoring contraction force in real time, providing a much more accurate way to screening drug effect for the heart tissues. So let me first describe the fabrication process of our 3D sensors. First, we laminated a PI film onto the PDMS subject. Then we deposit a thin metal layer onto the PI film, and then using the lithography and the etching process to create the sensor patterns. Next, we add an insulating layer and structure support layers. And then using the laser cutting to define the final sensor shape and release it. Finally, we attach a small iron rod with epoxy and assemble the sensor to the 3D structure. And we validate the sensor assembling process through both simulation and experimental result. Finite element analysis confirms the structure integrated of the 3D assembly, with experimental results showing the excellent alignment between the simulated result and the fabricated structures. To evaluate the sensor performance, the sensor resistance is configured as a Western bridge circuit and connected to the amplifier and the DQ system. Displacement was applied to the sensor's free end using a probe with a movement stage to simulate loading and unloading processes, and the output voltage change was recorded by the computer. In the first experiment, a probe was used to apply displacement to the free end of the sensor, simulating a single loading and unloading cycle, while recording voltage change in the bridge circuit. After loading and unloading process, the sensor returned to the initial state, demonstrating excellent electricity recovery. The string sensor exhibits a minimal detection accuracy of the time micrometer at a sensor tip. In another experiment, continuous displacement range from 0 to 900 micrometer were applied to the sensor's free end, simulating multi-stage string loading and unloading. This experiment confirmed the linear relationship between the string and voltage across different displacement levels, further validating the sensor's sensitivity and accuracy. Here I will explain the concept of magnetically controlled mechanical stimulation. In Figure A shows the displacement of a 3D sensor under electromagnetic control. The system uses a PWM with a 50% duty cycle to regulate the magnetic field by switching the electromagnet, which drives the iron rod on the sensor to simulate the mechanical stimulation. And Figure C shows under 1 Hz cyclic magnetic field. The string sensor's voltage signal accurately captures and reflects the non-contact cyclic mechanical stimulation. In addition to mechanical stimulation, we integrated a bioprinting system for heart tissue engineering. Figure A shows our bioprinting equipment, which consists of a pressure excretion, a temperature-controlled nozzle, and a temperature-controlled printing bed. Figure B outlines the process of printing collagen bioink onto the 3D sensor. First, the printing pathway is designed, and parameters are adjusted by software. Then, the printer deposits the bioink onto the sensor along the predefined path. The next step is to capture 3D engineered heart tissue on the sensors. The neutralized collagen bioink is mixed with a freshly isolated neonatal rat ventricular cardiomyocyte, and then loaded into the printing syringe. A ring-shaped structure is printed around the sensor. Over time, the tissue undergoes a maturation phase, gradually compacting and developing synchronized beating. This contraction force generated by the heart tissues causes the sensor to deformation, providing the real-time insights into its mechanical properties. As the capture time increased, the contraction and relaxation duration of the 3D engineered heart tissue gradually extended. This phenomenon may be attributed to the overactivation or the delayed activation of the L-type calcium channel in the cardiomyocyte during the prolonged capture, leading to the prolonged calcium influx and subsequently extended contraction duration. Such behavior aligns with the phenotypic characteristics of the long QT syndrome, suggesting that the 3D engineered heart tissue could serve as a potential disease mode for long QT syndrome. To validate the usability of this heart tissue as a disease model, verapamil was administered to assess its effect on this model. Verapamil, a calcium channel blocker, reduces the calcium influx into the cardiomyocyte, thereby decreasing the excitability and the contraction force of the cells. The result demonstrates that with increasing concentration of the verapamil, the contraction duration of the heart tissue decreased, and the beating frequency became more and more regular. This indicated that verapamil effectively shortened the contraction duration and improved the electrical synchronization in the long QT syndrome disease model. These findings highlight the potential of our 3D heart tissue as a long QT syndrome disease model and as a platform for drug screening applications. In conclusion, the organ-based 3D sensor platform has proven to be highly effective for monitoring 3D heart tissue. It not only provides highly accurate contraction force measurement with a markable detection limit of 10 micrometer, but also offers reliable and consistent cyclic mechanical stimulation through its advanced electromagnetic system. By using verapamil on long QT disease model, we demonstrate the platform capability for the cardiotoxicity evaluation, showcasing its potential as a tool for drug screening and disease modeling. Looking ahead, we aim to further develop this platform to study a wider range of the heart disease models and to test new drug candidates, paving the way for breakthrough in cardiovascular research and clinical applications. Collectively, this advancement positions our platform as a valuable tool in advancing heart disease research and treatment. Thank you for your attention. This is all my presentation. Unfortunately, I'm unable to take a question live, so please feel free to contact me via the email if you have any question about this research. Thank you very much.